Hello lovelies, in this video we're going to be going through kidney structure for your A-level biology. Now there's some really detailed diagrams in here that you're going to need to be able to label and then work out what each of the different parts of the kidney do. This is a favourite topic, and especially when they like to link to lots of other things, because it is a place, the kidneys are a place where lots and lots of things happen. If you want more questions on this, or if you want lots of revision questions to check that you're doing okay, then there are loads of these waiting for you over my website.
Hi everyone. So today we're going to look at kidney structure and function. So we need to be able to talk about the sort of overall structure of the kidney, going quite into detail about how the kidney carries out its function. So the kidneys are part of the urinary system, which contains the bladder as well. And so they're the three main organs in this excretory system that we have. They have two main roles, the kidneys, as part of this system. Osmoregulation, which is maintaining the water potential of blood, as we've said, and we've talked about why that's important as well. So they help to either remove excess water or they help to retain water if we need to. We're mostly going to talk about kidney in the singular as we move through this, but we have got two of them and they have exactly the same structure, you just have one on each side. They are fed in terms of blood and oxygenated blood by the renal artery, and then the waste and other things from the kidney and the kidney tissues are removed through the renal vein. The kidneys, as we said, act as filters, and they are removing urea from the blood and then diluting that with water to form what we call urine. Once the bladder is full, so it collects the urine in the bladder. Once the bladder is full, then the urine will leave the body out through the urethra, which is probably the term you've heard, and then ureter is obviously a similar type of tube, hence the name. The other thing the kidney does is reabsorb important amino acids, salts, glucose that it filters out of the blood. So the filtration process initially removes a lot of things, including urea, but we don't want all of that to leave the body because some of it is very useful. So we need to reabsorb that and make sure that that doesn't get lost in our urine. And that process is known as selective reabsorption. And then the other process is obviously the osmoregulation and to do with water as well. So we've got a couple of roles, and as you can see, we'll talk through as we move through the kidney how those happen. So let's start by looking at the gross kidney structure, so the overall kidney structure from quite far away before we kind of zoom in and see a bit closer. So we need to be able to label these parts of the kidney as well as kind of the smaller structural parts. So the first part, sort of the outer layer, not quite the very sort of brown layer that you can see the outside, but the first section that we can see here is called the cortex, the outermost layer of the inside of the kidney. And it contains the tops of approximately about 1 million nephrons per kidney. And these are the series of tubes that are repeated many times, up to 1 million times, that help us actually do the filtration and the reabsorption. It could be up to 2.5 million, I think, is the highest it's been. But there's a lot of them. And the top parts of those nephrons sit in this cortex layer. So this is one nephron here, highlighted in green. The top parts are in the cortex, and that is how we carry out most of the filtration and the absorption. And then the parts that extend down into the medulla are mostly used for osmoregulation and water control. You can see one of the structures here sort of taken out of the section of kidney, and we'll look at that in more detail as we go through figuring out the processes of reabsorption filtration. Now, the bottom part is the medulla, so the sort of inner layer of the kidney is the medulla, and it contains the tubes that drain the filtered wastes into the renal pelvis. So we can see here we've got some of those tubes of the nephron coming down all the way down through the medulla, and then on the larger diagram, we can see the renal pelvis is kind of like the collection of tubes in the middle to join up to the ureter. And then obviously, as we said, the ureter earlier, that is where the urine travels down to the bladder. So that's the overall structure. We need to remember the cortex is on the outside, the medulla is on the inside. Top parts of the nephron are found in the cortex. The bottom parts of the nephrons are found in the medulla. There's at least 1 million nephrons, there's lots of them, and all of those tubes drain into the renal pelvis towards the centre of the kidney, which collects all of those ducts together and then connects that to the ureter. Okay, so we're going to start at the beginning of the nephron with ultrafiltration. So ultrafiltration, as it says, we're filtering the blood. We're filtering out a lot of things from the blood, and it takes part in this section called the glomerulus. So the glomerulus is actually the knot of capillaries that are sitting inside this little sac that we call the Bowman's capsule. So this is the start of the nephron and where all of the blood first enters and where we start our process of filtering. We have an afferent arteriole. So remember, it goes arteries, smaller than arteries is arterioles, smaller than arterioles are capillaries. So this is an afferent arteriole, and as it enters the Bowman's capsule, it then narrows into capillaries that twist and branch all around each other, and then they come back out as the efferent arteriole. So the afferent arteriole has a wider lumen than the efferent arteriole. So that means that a larger volume of blood can come in, and as it goes through the capillaries and then you've got a narrower lumen coming out, that forces it and makes it at high pressure because less volume of blood can actually leave compared to what enters. So that creates a high hydrostatic pressure in the knot of capillaries, and that forces all the plasma from the blood through the walls of capillaries. And so this is where we get off the stage of filtration. So we're forcing out the blood plasma and small molecules that are contained in it out through the walls of capillaries. This is very similar to tissue fluid, works in the same way. And again, what's filtered out is very similar. So it's small molecules, things that are dissolved in the plasma that are small, that can fit through those walls of the capillaries. So things like glucose, amino acids, urea, obviously, we're going to be removing that mostly, water, ions, etc. So these are then being pushed out because they're dissolved in the plasma and they can be forced out of those small holes in the capillaries, and then they enter the next part of the filtering system. What stays in the blood? Well, large proteins stay, and large cells, including red blood cells and anything else that can't pass through those small holes of the capillary, works exactly the same way as in tissue fluid. However, this is obviously an important step because if there is then anything, such as large proteins or red blood cells, so blood in the urine, then part of this system is broken and this filtration system won't be working correctly. And that can help us diagnose various problems. So the fluid that's forced out is forced out into the lumen of the Bowman's capsule. So the Bowman's capsule is this whole structure that sits around the glomerulus, and it's kind of like a cup-shaped sac that then forms and joins up to the rest of the nephron tubes. So if the filtrate is forced out of the capillaries, it sort of gathers in this sac, and then it's going to run down and filter into the next part of the nephron, which is the proximal convoluted tubule. Now, you may have noticed these kind of weird sort of centipede-looking cells around my capillaries. They are known as podocytes, and they're specialised epithelial cells of the Bowman's capsule. So this kind of layer of the Bowman's capsule that juts up against to the capillaries are actually these specialised cells, which have these kind of foot-like processes. That's why they're called podocytes, comes from the word similar to the word ped, like foot. And they wrap around the capillaries and they create this extra layer of filtration. So it's not just whether something can fit through the holes in the capillaries. It's also whether it can fit between these slits that are created where these protrusions of the podocytes kind of jot up against each other. And also these podocytes secrete what's called the basement membrane. So it's a membrane that, again, is kind of the layer of tissue that is up against these capillaries that fluid would have to pass through to get into the Bowman's capsule. And it's kind of like a mesh of proteins. And so this filtering system stops, again, any large molecules or charged particles from entering the glomerular filtrate. So anything big or superpolar wouldn't be able to get through this basement membrane and then get through the filtration slits between the podocytes. And so anything else that does is something that's supposed to be in the filtrate. So once all of that fluid has been pushed out through all of those layers of filtration, what's left is now known as the glomerular filtrate, and it is going to pass sort of into the Bowman's capsule and then drain down into the next stage of the nephron, which is the proximal convoluted tubule. Proximal meaning next to, convoluted meaning twisted, windy, like a windy, long, complicated story, and a tubule, so a tube. So it's the tube that is next to the glomerulus, and it's kind of twisted. So the next stage, once we have filtered out all of the substances, we actually need to reabsorb quite a lot of them. So as we said, the kidney makes sure that we don't lose a load of these really essential nutrients, and we only are filtering out kind of waste substances like urea that we don't want to keep. So selective reabsorption is what happens. And as the glomerular filtrate moves through the proximal convoluted tubule, about 85% of it is reabsorbed into the blood, which is quite a lot. but that makes sense if you think about all the things that are going to be in there that we actually need. So all of the glucose is reabsorbed because it's needed for respiration. All of the amino acids are reabsorbed because they're needed for protein synthesis. Some ions are reabsorbed, mostly ones that are normally in low concentration in the blood and that aren't commonly found in the diet, so not easy to replace. And most of the water just moves back in by osmosis. So the cells lining the PCT are adapted to maximise absorption, and they have adaptations that are similar to the cells that line the small intestine. So you can see them here lining that first section that we can see of the PCT, just coming straight off of the Bowman's capsule. So what adaptations do they have? Well, they have a folded membrane, which obviously gives a large surface area, speeds up rates of diffusion, but also it allows lots of reabsorption to continue at the same time. You have lots of membrane proteins, so channel proteins and carrier proteins, which allow us to do lots of facilitated diffusion and co-transport as well. So we're going to need that for glucose and amino acids can both be moved by co-transport. Lots of ribosomes, or potentially a large rough endoplasmic reticulum, which is going to help us to make all of those membrane proteins that we need. Lots of mitochondria, obviously to carry out respiration, to provide the ATP that we need to do the protein synthesis and the active transport. So this is a cell that you probably potentially need to talk about as a cell that has specialised adaptations, or a specialised cell. They might ask quite a long answer question about how cells of the PCT are adapted, and these are the things that we'd need to talk about. They could give you this just a same thing you would say about an epithelial cell that lines the lumen of the small intestine. Basically, any cell that's doing a lot of transport is going to need all of these adaptations. Okay, so we're talking about the reabsorption of water now. We've talked about filtration and then selectively reabsorbing the nutrients that we need to keep in the body because they're important, and now we're looking at the reabsorption of water. So the function of the loop of Henle is to start off this process, and its purpose is to create a really low water potential in the medulla of the kidney. So remember, up until this point, we've been looking at the top part of the nephron, so the glomerulus and the PCT, and all of that is located in the cortex. And the rest of the loop of Henle that hangs down, and also the collecting duct that hangs down, are in the medulla. And that's important because there's a separation there of tissues. And we're focusing on the tissue in the medulla because that is what we're going to change the water potential of in order to encourage water to leave the filtrate by osmosis. So the descending limb, descending as in moving down, it doesn't necessarily mean moving down here. What it means is moving down into the medulla or away from the cortex. And limb here just means like arm, basically. So the descending limb is moving down into the medulla, and the ascending limb is the filtrate moving back towards the cortex. Salts are going to be transferred, so ions are going to be transferred from the ascending limb to the descending limb, and the concentration of the salts in the medulla is then going to increase, and therefore it's going to decrease the water potential of the tissue that surrounds the loop of Henle. It decreases as you go down, and the lowest water potential will be here, towards the apex. Water's then going to follow this, so through osmosis we've created a really low water potential in the tissue outside of the loop of Henle. Water will follow it out down the osmotic gradient, and because you can see there's this network of capillaries wrapped around it, that will eventually make its way back into the blood. We're going to look at that in more detail in a second because you need to be able to explain exactly how that happens, but let's just have a look at what happens after it comes back up the ascending limb. So it's going to enter the distal convoluted tubule, distal meaning kind of further away, again same, convoluted, twisted, you can see it's twisted tubule. And this is another part of the nephron, it's in the cortex, and it just is an opportunity for more water to be reabsorbed into the blood by osmosis at this point, if there's still water in the filtrate, but that can move by osmosis into the blood. Finally, the filtrate enters the collecting duct, and the collecting duct here is going to collect, as it says, all of the filtrate from sometimes multiple nephrons, and all of that is going to be channeled down then to the renal pelvis, where we're going to collect all of that and send it to the bladder as urine. So the permeability of the membrane of this part of the nephron, the collecting duct, is controlled by the hormone ADH, or antidiuretic hormone. Antidiuretic hormone is released by the pituitary gland in the brain, and it controls how much water enters the urine. We're going to look at how it controls that in a second, because that is our osmoregulation and how we control the water potential of blood. So we need to be able to explain how the loop of Henle actually works in order to concentrate the urine and reabsorb the water. So the loop of Henle is what we call a hairpin countercurrent multiplier, because what it does is it allows the concentration of the filtrate to be increased by reabsorbing water, and it also increases the concentration of the tissue around the loop of Henle in the medulla in order to help to remove the water, which we'll explain in a second, at the same time. And it looks like a hairpin because it's this kind of U, sort of thin, long U shape. So overall, this mechanism is important because what it does is allows the nephron to reabsorb more water than it would otherwise, and it allows it to concentrate the urine as well, and it uses less energy overall. It's actually an energy efficient way of doing this. Okay, this can seem quite complicated, but we'll go through it step by step, and it should hopefully make sense. So we start with the ascending limb. So remember, that's where fluid is moving towards the cortex. So towards the edge of the kidney, or towards the top of the nephron. So as fluid is moving up the ascending limb, sodium and other ions, so sodium, potassium, chloride ions, are actively transported out of the filtrate into the tissue fluid. They're normally moved out by diffusion at the base, and then as they get fewer ions left in the filtrate as we move up the ascending limb, then we have to start using active transport. What this does is by moving the ions out, is that it lowers the water potential of the surrounding tissue in the medulla. So that's important because what we want that to do is we're going to need that in order to move water out of the filtrate by osmosis. But it doesn't follow the ions on the ascending limb because that is impermeable to water. So the membrane of the loop of Henle on the ascending limb is impermeable to water. So water can't move out by osmosis at that point. So where the ions have moved out into the tissue fluid and the tissue of the medulla, then we have a low water potential. And those walls of the ascending limb are impermeable to water. We have to think about how this is moving. So there's more ions leaving at the base of the loop of Henle because they're diffusing out. As we go up the ascending limb, there's fewer ions, which is why we're having to actively transport it. So more are being lost from the bottom of the loop of Henle. So the lower you go down into the medulla, the lower you go down towards the point of the loop of Henle, the lower the water potential. So the water potential decreases as you go down further into the medulla. And this is important because the filtrate coming in at the top of the descending limb is going to have the most water. And so as it moves down, it's going to lose water by osmosis. And so we need to have this concentration gradient where we have a high water potential at the top of the descending limb of the loop of Henle and a lower water potential towards the bottom of the descending limb of the loop of Henle to make sure we maintain that concentration gradient of water. So there's always more water in the filtrate than in the tissue fluid outside, and that's going to guarantee that our water is going to leave the descending limb and go into the tissue fluid and then ultimately be absorbed into the capillaries that, remember, are surrounding this loop of Henle. I've only got one sort of in the middle here, but you'd have this kind of a web of capillaries surrounding the loop of Henle, and so the water would ultimately move back into the blood. So let's think about what's happening here and just sort of look at it overall, because we've sort of done it backwards because it makes more sense that way. But we started with a lot of water and a lot of ions at the start of the descending limb. As we move down the descending limb, water is lost due to osmosis because the walls of the descending limb are permeable to water, so it moves out into the tissue fluid surround and ultimately into the capillaries because it's following its concentration gradient, because the tissue fluid of the medulla has a very, very low water potential because of all the ions that are in it. When we get to the bottom of the loop of Henle, then we start diffusing ions out. So sodium, potassium, chloride ions. So not only have we lost a lot of water by this point, we're now also losing ions. So although we've lost a lot of water, we still have a lot of ions at this point, at the very apex. So that's where we have our lowest water potential of the filtrate because we've lost the water, but we've kept the ions. Once the ions also leave, actually, at this point, we have a slightly higher water potential again because we will have some water still left and we've also lost ions. So if we're removing ions, that increases the water concentration and decreases the solute concentration. Does that make sense? So ions are leaving, and so ions leaving will always increase the water content. So we've managed to reabsorb quite a lot of water without needing to expend much energy. Most of it is through diffusion of ions at the bottom of the loop of Henle. we have used a bit of energy for active transport, but because there's no water being able to leave the ascending limb, it's just ions, then we don't very quickly create an equilibrium with that water. It only moves out across the other side of the descending limb. I hope that makes sense. It can be quite complicated, but if you follow the steps and those bullet points 1, 2, and 3, and that's how you explain it, for example, in a long answer question. And you could always sketch yourself out a little quick diagram to remember descending, permeable to water, ascending, impermeable to water, ions being actively transported out at the top of the ascending, diffusion of ions down the bottom, water moves out of the filtrate as it moves down the descending limb, because as you move down the outside in the medulla, the water potential gets lower and lower and lower because more and more ions are being let out and diffused out or actively transported out into the tissue fluid. So it moves down its water gradient as osmosis happens. And then as the filtrate is coming back up, so remember we haven't lost it all, as the filtrate, whatever's left, comes back up, it is losing ions. So that means the water potential by the time you reach the top of the ascending limb isn't as low in the filtrate as it is at the bottom of the loop of Henle. And remember, when we're talking about water leaving, it does leave and it will go into the tissue fluid, but remember the capillaries are surrounding the loop of Henle, so the water will eventually be reabsorbed into the blood, which is the point, and that's what we want. So any change in blood water potential is detected by osmoreceptors in the hypothalamus. So osmo coming from the same root as we talk about with osmosis. So this idea that they're detecting water potential of the blood, and they're located in the hypothalamus, which is a part of the brain we've already mentioned because that's where our temperature control center is. So if the hypothalamus detects that blood water potential is too low, for example, it's going to stimulate the posterior pituitary gland to secrete more of a hormone called antidiuretic hormone. So the posterior pituitary gland is here. You can see our pituitary gland is labelled. There's two parts that kind of hang down. So this is the anterior, and then this one is the posterior because it's towards the back. So in this case, the hypothalamus is where the receptors are. It's not actually releasing any hormones that are going to bring out a change. So it's actually communicating to another gland. So it's acting almost as the coordinator here. It's sending another signal to a gland that's then going to release a hormone, and that hormone is then going to have the effect. So that's the kind of slightly exaggerated sequence that we've seen before with these kind of like the hormone sequences that we've looked at. So osmoreceptors, osmo coming from the same root as osmosis, they are detecting a change in water potential. And then antidiuretic hormone is the hormone that's going to be secreted. You might have heard that there are some drinks that are known as diuretics. And so things like tea, for example, is a diuretic. And what it does is that it acts in the same way as antidiuretic hormone can have, in that it actually causes you to lose more water in your urine. So you'll sort of wee more because instead of reabsorbing that water, you are losing it in your urine. So it works in a similar way to how we're going to say ADH works. And so some of those drinks and various other things have this diuretic ability. And we say that antidiuretic hormone has got the anti in front of it. It can do both. It can cause more water to be lost in the urine and less water to be lost in the urine based on what the body needs. Okay, so let's just have a kind of summary of sequence of events to do with how we increase or decrease our blood water potential, and then we'll have a look in a bit more detail about how ADH actually causes this change. So blood water potential increases away from the optimum. Osmoreceptors are going to be able to detect high blood water potential, and they're going to then send that signal to the posterior pituitary gland. The posterior pituitary gland is going to secrete less ADH or stop secreting ADH altogether. This means that less water is going to be reabsorbed into the blood, which means we're going to lose more water through urine. This makes sense. If we're not removing it back into the blood, so stopping it from leaving the body, then more of it is going to leave the body in the urine, which is what we want, because ultimately we get rid of more water from the blood. That's going to decrease that blood water content. So let's look at it the other way. If blood water potential drops to quite low water potential, this is obviously detected again by the osmoreceptors in the hypothalamus. The posterior pituitary gland will be stimulated this time to release more ADH or start releasing ADH. This is going to cause more water to be reabsorbed into the blood. So we're retaining more water. We're not letting it leave the body through the urine. We're reabsorbing it into the blood to try and bring those blood water potential levels back up. So you can see how with less ADH, it means you're going to wee more. You'll have more volume of urine to wee. Your bladder will be fuller and you will remove more water. If you have more ADH, you'll actually not wee as much. You will have low volume of urine passing out because we all have reabsorbed the most of that water. So how does ADH actually affect how much water is reabsorbed into the blood, or how much water ends up in urine? You need to be able to explain both scenarios and do it kind of as a process as we've talked about. So what happens when ADH binds and then what that chain reaction that it causes. And it works in a similar way to how we've seen before, using that secondary messenger cascade. Cells in the wall of the collecting duct have receptors for ADH, and they also contain vesicles, which have aquaporins in them. So aquaporins are just channels, protein channels, but specifically for water, okay? So here I have my cells. Okay, so if there's too little water in the blood, there's not enough water in the blood, ADH is going to bind to the receptors on the collecting duct cells. So here's my ADH binding. And then that causes, in the same way that we've talked about all the other things happening using that secondary messenger cascade, it causes vesicles containing the aquaporins to move towards the membrane where it's joining the collecting duct. Okay? What that does is that's going to increase the cell's permeability to water. So it makes it more permeable to water. This results in more water leaving the collecting duct through these aquaporin channels, and then it will move out of those cells and into the bloodstream. So it's going to get reabsorbed into the blood. But most importantly, the water is leaving the filtrate and it's not going to be in the urine. So we create a smaller volume of more concentrated urine. And it's more concentrated because there's less water in it, because we've removed the water and reabsorbed it. Obviously, if we think about the opposite, then the opposite happens. So if there's too much water in the blood, less ADH binds to receptors. So there's no ADH or very little ADH binding to receptors at all. The vesicles containing the aquaporins do not move, and so they don't fuse with the membrane. So this makes the membrane of the cells slightly less permeable to water. And so less water leaves the collecting duct and filtrate and goes into the blood. And so we create a larger volume of more dilute urine, because more water is going to stay in the collecting duct and more water will end up passing down through and into the urine. And because we've got more water, that dilutes the urea and the other salts, and we create more dilute urine. Okay, so that is the kidney structure and function summarized. It's quite a long one, but I think it's good to do it all in one go. And so we need to know, sort of like we said, the growth structure, then go more into detail about the nephron structure, talk about how ultrafiltration happens at the glomerulus, talk about how selective reabsorption happens at the proximal convoluted tubule, then talk about how the loop of Henle works and explain that, then talk about how we ultimately can control how much water gets reabsorbed at the collecting duct using ADH, which is another one of our control mechanisms using these hormones and the endocrine system.
Ouch! This is why in some videos I've explained scratches.